Hello everyone, this is Mrs. Travai, and today we're going to be talking about PCR. These PowerPoints have been provided by Rutgers University and the Waksman Student Scholars Program. So we're back to this picture here, and we're going to take a look at how to identify how large the insert is from the bacterial culture. If you remember, we did the PCR from the bacterial culture, so we're looking at the actual size of the insert at this point. And how can we determine that? Basically, in our plasmid, the gene has been inserted and ligated, and this area of the plasmid is going to be amplified so that we can see how large our insert is. So the principles behind PCR are pretty simple. A specific fragment of DNA is repeatedly synthesized or copied. We amplify that section using a DNA polymerase. And basically what we end up with is a millionfold or more of the actual target sequence or the area that we want to amplify, which can then be sent for sequencing. Or we can proceed through our processes and we can mini prep the DNA if it's large enough, and then do a restriction digest in this program, and then we'll send it out for sequencing. This process was created or perfected really by Carrie Mullis. Really an interesting person in science. There's a link there that you could take a look at. But he now travels the world and does a lot of speaking engagements and things like that. He's an interesting person. And basically, PCR stands for polymerase chain reaction, and here are just two examples of how this process is used to either to look at specific pieces of DNA or sequencing, or to amplify a piece of DNA for sequencing in different types of forensics cases. In the first one, a person's cleared from murder, and this has happened quite a bit in the last 15 years, where they've actually looked at reopened cases and taken a look at the evidence that wasn't analyzed initially. So let's take a look at the steps in the polymerase chain reaction in PCR. There's a great animation at the DNA Learning Center, and I've linked this on the EDU 2.0 site. And if you could take a look, I've also placed it on the Google Classroom, but if you could take a look at it, it goes through how this process occurs and how we end up with such a huge amount of DNA at the end. Basically, you're going to denature the DNA at about 95 degrees centigrade, and we're going to add primers. And when we add the primers are already in the tube when we put them into the thermocycler. And when we add the primers, we have to lower the temperature so that they will anneal at about 50 degrees. And these temperatures can vary ever so slightly depending on the reactions and the DNA you're trying to amplify. Basically, we extend the chains. We then warm up the thermocycler, changes temperature to 72 degrees, and the Taq polymerase will then take the nucleotides and add them to the template strands in the area where the primers are. So we repeat the steps, and in some cases we'll repeat it about 30 times, which will end up with over 1 million pieces of DNA. So here's your first clicker question, and again, you can go on to the quiz and enter the answers. So you can pause the video and try your best to see if you can answer this question. So in this area, and I've changed it here to represent the Landoltia DNA, there's a polylinker region where the Landoltia DNA was added. This is the forward primer, and we'll go back. The forward primer and the reverse primer down here, this is the area that we're going to amplify. And so it's not just our DNA, we're actually amplifying part of the vector, our plasmid that we are using. And so we're going to have some extra sections in the insert or the fragment when we actually take a look at our gels. That extra section is the area where the primers were bound, and it's going to be roughly about 200 bases extra. So this is just reviewing what we've already done, where we grew the colonies and we labeled our tubes. And remember that when we do PCR, you're going to take your overnight culture and you diluted it with water. because you need only a little bit of DNA to begin this process. If you have too much DNA, you'll actually use up the nucleotides before you amplify and before you get through too many steps of the process. You want to make sure you have very little DNA to work with initially, or a dilute sample, and that way the process will go on as expected. So we're going to be making a reaction mix. And the nice thing is that if we do have the PCR beads itself has Taq buffer, has the nucleotides, and the Taq polymerase in a freeze-dried bead. Very convenient, very easy for us to use, but a little expensive. And then you're going to add the primers and your DNA to make a total volume of 25 microliters. Okay, so try your question at clicker question number 2. You're going to prepare this reaction, and typically you'll prepare a 5-reaction mix or a 5X mix, and that 5X mix is going to be enough for 5 reactions, but I typically tell students to share it with 4 students because you want to make sure you have enough in case there's a slight error in pipetting. So when you make the 5-reaction mix, you're not adding the DNA, you're adding water and the 2 primers, the forward and the reverse, into a 5-reaction mix tube. You're then going to tap that to mix, and each member of your team is going to take 23 microliters and put it into their PCR tube with the bead. Okay, now after you aliquot the mix into your PCR tube, that's the little tube. Now the PCR tube is very different than the micro tube. The microcentrifuge tube is thicker plastic, and it's larger in size, and that's typically what we use to make our mix, dilute our cultures, and for other types of experiments that we will be doing. The PCR tube is unique where the plastic is thin, it's a smaller tube, and it allows heat to transfer through the tube quickly, allowing the process to happen efficiently. So basically you're going to add, the last step is where you add 2 microliters of diluted bacterial colony to your PCR tube. Again, don't over pipette this section. Try to pick up only 2 microliters, but please, when you do this step, look at the tip before you take your dilute culture, and then look at it once you've obtained the culture to make sure that you have something in the tip. Okay, you're going to give the tube to the teacher as soon as you're done, and I'm going to put it into the thermocycler. Okay, the primers start to attach the nucleotides. If you don't start the reaction right after you add the DNA to the mix, then you really are going to start to reduce the amount of target sequence that's amplified. It's just going to start adding nucleotides. So we want to actually denature the DNA and make the reaction work the way it's intended. Okay, so here is basically the temperature ranges and what's happening. I would definitely know this. You're eventually going to have a test or a quiz, and this is one of those, this information is important to understand what's happening at each step. So you need to understand that the initial is denaturing the DNA, then you anneal the primer, and then you elongate the strands. We actually do an additional elongation to ensure that all the DNAs are at full length. And the last step is to actually put it in a 4-degree hold and keep it cold overnight. And it's nice because our machines do that, and we're able to obtain the samples in the morning, and they're nice and cool. The next day, now that your sample's amplified, we can add loading dye and run the samples in a gel.